Today we've got an esteemed and knowledgeable panel on gene therapy and some of the challenges around it. We're certainly going to be discussing some of those today, and I'd like to try and get into a number of different topics here. Some of them to do with the challenges of redosing an AAV, some of them to do with what we're doing with dealing with patients with high neutralizing antibody titers and dosing for the first time or perhaps redosing. Ways of looking at where the field is moving today with respect to new AAVs and ones where we can change the tropism to be more targeted for muscle and certainly less targeted towards liver. And beginning, I think, with our deep understanding today of some of the immune issues and the safety issues that are associated with this. Karsten, I'll let the group introduce itself before we actually start, but I think that there's some of the topics that I'm going to try and cross over today. So let's start off with introductions.
Good morning. My name is Sharif Tabe Bordbar, and I'm the Chief Scientific Officer of K Therapeutics.
I'm Karsten Bornemann. I'm a pediatric neurologist, physician scientist at the National Institutes of Health, working in neuromuscular disease and gene therapy.
I'm Kevin Flanagan, Director of the Center for Gene Therapy at Nationwide Children's Hospital, and work on gene therapies and other approaches for muscular dystrophies.
Hello, I'm Barry Byrne. I'm at the University of Florida, just a few miles up the road, and I'm the director of the Pal Gene Therapy Center there. My clinical background's in pediatric cardiology.
I'm Robin Van Poelheist. I'm a medical director at Sarepta.
And thank you all for being here. So, Karsten, let me begin with you in that we have recently seen the published material out of the European workshop for reviewing some of the safety assessments and certainly the recommendations associated with gene therapy. There was quite a panel of both the industry representatives as well as academics that were involved in that. And could you take us through some of the topics that were discussed and the recommendations that were made?
Yeah, this all originated around an initiative that we were able to put together when the first emergent side effects arose around anti-transgene immunity. And this was happening in 3 of the 4 trials at the moment that were at the time were happening. Just to maybe bring the room up to speed, what happened there is that in rapid succession, several boys that were dosed with various forms of the microdystrophin in different trials with different AAV capsids had a very similar adverse event, which was an inflammation of the muscles and the heart muscle that arose several weeks after dosing. That happened across these different trials. And Francesco Montoni at UCL had an overview of these different trials and engaged myself as well as all the sponsors, Sarepta, Pfizer, Genethon, Roche, Solid, to see whether we couldn't come together rapidly to try to understand these adverse events that were happening across the trials. And it was really very encouraging to see that there were no boundaries. This happened in real time. People were coming together. We engaged experts such as Kevin Flanagan as well to really try to understand this quickly to see what was happening and how that impacted safety and the way forward in these gene therapy trials. And what was very clear very quickly is that all the boys who had these adverse events had genetic deletions in their genome that were overlapping with transgene sequences. So what that means is you learned this morning that the protein information is encoded in the genome, and the most common mutation in Duchenne muscular dystrophy is a deletion, so a loss of genetic material. When you lose genetic material that encodes dystrophin, you cannot make that protein that's encoded in the deleted region. So your immune system, your body has never seen that protein. And if that protein is part of the transgene that is supplied as a gene therapy, that protein will now be novel to the immune system, to the body. The body has never seen it because the information for that is deleted in the boy. So basically, the boy is immunonaive against the transgene protein. And the immune system doesn't ignore that. The immune system sees it and then started to reject the transgene because this was now recognized as foreign, as something that hasn't been seen by the boy's immune system before. And so that happened to the degree that the immune system now attacked the transgene, and with the transgene, the muscle where the transgene was expressed, and the heart. And that led to loss of ambulation, to the need for ICU care. Fortunately, no boy died in that situation, and they recovered with immune suppression. But it raised the specter that gene therapy, if you do gene therapy in a situation where the immune system is not aware what's coming at it, that this could be leading to rejection. Similar to organ transplantation, where you transplant an organ that's foreign to the body and the immune system reacts to that. And that led to redefining really what is the most offensive region in these transgenes. And it came down on one particular region of the dystrophin protein, the hinge-1 region, which is particularly immunogenic. And if the boy is deleted for the protein information for that region, but it's supplied by the transgene, this can happen. And so that led to the current exclusions that you all know in the application for dystrophin minigene gene therapy, and we have to discuss what the way out of that is. But it led to this cross-sponsor initiative to really discuss this, bring it to the table, show the data, share the data, and discuss reasons why and potential ways forward to prevent this from happening, and also in the future allow dosing for boys that are deleted in that region. And so the ENMC workshop was one outcome for that. So we discussed all of these issues. We discussed ways of monitoring, ways of preventing this from happening, and potential strategies around it, and also tried to understand other implications of AAV gene therapy and its safety, and how to form collaborative initiatives to really understand as a group going forward how to monitor this, how to react to it, how to prevent it, and how to make gene therapy safe.
So it's got implications certainly for clinical trials, and we see that in the exclusion criteria. What's the recommendations moving forward as these drugs are approved and out of a clinical setting in order to deal with the broader population?
For the deletion, particular deletions, yeah, so I invite other panelists to talk about this as well. At the moment, the strategy is simple avoidance of the most. So it doesn't, not every deletion is made the same way in its immunological risk. So there's some deletion that we are now homing into, in particular these around hinge-1, which is this 8 to 11 encoded, that are particularly immunogenic for many reasons. One is that these peptides in the protein are actually quite exposed. They're literally on the surface, and so they're very visible to the immune system. And the second part of that equation is everyone is different in the makeup of their own immune system. We're as individual in our immune systems as we're individuals as personalities. And so we have to understand whether there's certain individuals who are more prone to expose these antigens and have this immune reaction, and others are maybe less likely. So that's the host part of the equation, if you will. And then there are other deletions and other proteins that are maybe not as offensive, so they're kind of more muted in the immune response. But if you have an individual, like a boy who received the gene therapy who is particularly prone to have immune reactions, that needs to be understood as well. So the way forward is to gradate and understand the relative risks of these genotypes, of these deletions, and the immunogenicity of it. And then develop, I think, as a group, as a field, we have to develop protocols that allow us to modulate the immune response proactively and monitor that. And that's not, I mean, it's not rocket science, if you will. We've been doing a transplantation for decades, and we know how to modulate the immune system, how to keep it in check, how to teach it to accept a new protein, a new antigen, how to develop tolerance against it. That's a magic word, tolerance against it. And so there are strategies to induce tolerance. There are even strategies to induce tolerance ahead of time. There are certain parts of the body, if you express a protein, for instance, in the liver, the immune system is much more likely to accept that protein as its own and develop tolerance against it. And so these strategies are all on the table, and I hope they'll be put in place as a field and then monitored for their effectiveness. I think we have to work together to kind of decide which protocol is the one to go forward in terms of the immunomodulation, who's going to go first and do it, and how to monitor it. But I'm a firm believer that can be done and should be done relatively soon. I don't know, see what other people...
I'll invite Barry and Ketan to come in. You've dosed both Duchenne as well as many other patients in the field, and what's your thoughts about that expanded understanding of how we do immune modulation?
Yeah, so Karsten mentioned the analogy to transplant, and we wrote a commentary several months ago about how the relationship between gene therapy and the early period of transplant and what was learned to prevent graft failure. And I think we are applying some of those principles in studies we've done thus far, and we'll continue to refine the approach to minimize toxicity, promote long-term expression of the transgene, and ultimately enable gene therapy products to be used like other drugs, where they may be titrated to a certain level of effectiveness, where in the musculature, as we potentially treat earlier patients, as Kevin showed, really remarkable results in this youngest boy in the DUPE-2 trial, that really challenged us to think about early treatment with the advent of newborn screening. And that's a really important concept, as long as it's linked to the notion of how to sustain that effect over the lifespan. And the biology of muscle is really against the notion that this would really be only once in a lifetime. And so we can really contemplate how to manage the immune responses to take advantage of the transformative nature of gene therapies, but that can really last into adulthood. I don't know if you want to comment, because that was somewhat unexpected, really, in that patient. So it was really been a pleasant surprise to see both the durability that results from the homogeneous expression of a full-length dystrophin in the DUPE-2 study.
Yeah, there certainly raises questions of durability. Mike, I don't know if you want to come to those now or later on, but you know, the issues of durability touch on immune responses, certainly, in some ways. Durability is a broader question, I think. We know with AAV in liver-directed diseases, we know that AAV genomes can express for a long time, a decade or more. But what does durability mean in muscle? When we treat someone small, where we're still going to have growth of muscle mass, what does it mean where there's ongoing degeneration of muscle and loss of genomes? We know that because CK is not perfectly normal in everybody. We know there's continued muscle breakdown. I'm optimistic, like Barry, in the example we showed, expressing a very highly functional, very highly protective protein probably will lead to a more durable response than some of the versions of less protective proteins. Yeah. And then long-term immune responses deteriorating, attacking muscle, expressing these transgenes, it ties in very closely with the durability question, not only safety, but durability.
Very much so, and you know, one concern that we just have to address and monitor is, so in these boys who had these adverse events, that was very clinically evident there. They had massive muscle inflammation, they lost the ability to walk, they needed to be immune suppressed. There was no question that this was going on. But of course, the question is, could there be, in the boys who don't have this, could there be underlying kind of low-level immune reactions, immune rejections going on over the years, and could that influence durability? Because that's something we can do something about. As we understand it, this can be treated and modulated. We just, I think, have to look very careful now that we know this is on the table, this can happen in gene therapy, that we make sure that we set everything to optimal, that the expression of the transgene is unhindered over years and not being shut off at low level by a kind of subclinical immune rejection. That's still something that we need to, as a field, address and kind of make sure that we do everything possible that the durability is as good as it can possibly be. The challenges in this disease are great, and so we need every little transcript to be as long as possible.
Can I add, I think that's one of the challenges to get this information, I think, for parents who've received gene therapies or will have received gene therapies. There will be a rationale, I think, as it goes along for us as clinicians and clinician scientists to say, to come back to parents at some point and say, we're going to ask you, can we biopsy your boys at some point later on to get these answers to these questions, to look at the relationship between inflammatory responses in muscle over years and expression over years. even after clinical delivery of it. So I don't say that we're asking to do that today. I just want to be able to say to parents that I'm quite sure that we will at some point be making those requests with good scientific rationale to understand these issues.
Yeah, what's your guesswork on durability? Like, you're following a young man who was dosed at 7 months. Where do you think we'd expect to see that effect reach out?
Well, I don't want to speculate too much. It's an N of 1, and Dr. Waldrop is really responsible for that trial. We have discussed in that example of what about it, and discussed with the DSMB, in fact, at what would be an optimal point to consider a repeat biopsy for this. That's an unexpected response. I think all of us would agree we didn't expect to see that kind of response. We want to, there will be a point where we're going to ask that family to repeat a biopsy. The scientist in me says, let's do it every year and so on. And don't worry, parents, I'm not saying we should do that. That's the scientist part. So it's a really decision about what seems to be reasonable about it. I would expect, in that particular example, when that child gets to be 5 or 6 years old, we'll be asking
yeah, that might be the case. I do recall the work in the dog, where it was the effect was long-lasting for the lifetime of the animal. But it's going to be really interesting following the young 7-month-old man.
Right.
So I was going to say another lesson learned from transplant is that when immune management was applied reactively, as is really happening today in the context of gene therapy, this led to a higher rate of graft failure until the practitioners in that period, which is in the mid-'80s, learned to use these drugs preventatively.
That's
and
the key.
It's much easier to manage, and the upfront exposure, which is really the biggest problem in the way gene therapy is administered. It's not administered a little bit at a time over a long time. It's this large exposure. So we have tried to replicate that approach in the study we just published, in which there's a period of coverage that lasts about 6 months, and then we can taper that immune management to allow both safety and efficacy to be optimized.
Okay. So extending the discussion a little bit out of the risk associated with gene therapy, there's certainly increased risk we recognize in individuals who carry neutralizing antibodies who are seropositive. And I know there's a number of studies going on, some in Sarepta, Robin, some that you're doing, Barry, with Vyvgart or with plasmapheresis and imlifidase. Could you comment on that and the likelihood of that to help patients who are outside of the range for dosing and how one can bring them down? Or how
about the
that might even
imlifidase study?
impact redosing?
Sorry, I just want to make sure I understand you. There's a lot of echo here.
Oh, there's the echo?
Yeah.
Yeah, really what's going on with plasmapheresis at Sarepta, what's going on with imlifidase, and
Okay,
for
sure.
Barry, it's really what's going on with Vyvgart, for really to educate patients about those that can't get into clinical trials, who are seropositive above a certain level.
Sure. So we are definitely committed to not leaving any patients behind with our gene therapy. So we do have ongoing research with the plasmapheresis and the imlifidase approaches. So far we have just published data with a non-human primate or a monkey study this year. And what we did there is we administered the gene therapy to the non-human primates. And then they were given 2 or 3 rounds of plasmapheresis, which is, just in case any of you aren't aware, plasma exchange will remove the antibodies from the plasma. And so the animals were given another administration of gene therapy after that. And so we found this did not result in any new safety signals and was well tolerated. And so we are excited about these results. And we are hopeful that this can lead to future work with patients that have pre-existing immunity, and also potentially serve as a launching pad for sequential dosing of gene therapies. For the imlifidase studies, we are looking at, and this is an ex-US study, so we're looking at imlifidase, which is an enzyme that breaks down antibodies. So this is kind of a different approach than removing them. So what we're doing there is we're going to be administering imlifidase, and then we're going to be checking for existing immunity or circulating antibodies prior to gene therapy administration 48 hours later. And so as long as after the imlifidase is administered and there's not immunity against our gene therapy vector 48 hours later, then the patients will receive our gene therapy. And so again, this is in Spain, and this is not currently ongoing, but it will be very soon.
Vivgard?
So we have had some clinical experience with plasmapheresis in patients who were part of other studies, and found that one of the challenges of this acute removal of antibodies is that the cells that produce them are still present. So there's an opportunity to use that methodology potentially paired with other approaches that use antibodies themselves as a therapy to remove those cells that make antibodies. So we have so many now antibody-derived therapies that are used in immunological diseases very successfully, that we're really repurposing some of those drugs to use in the context of Duchenne. So when there is a high sustained antibody level, and we know that these are actually now, thanks to some very early volunteers in gene therapy studies, actually high sustained antibodies are expected to persist over the lifespan. We've studied now about 400 patients, 115 of which were antibody positive over the last 5 years, and there's essentially no change. And then one individual who was a participant in a study 20 years ago remains antibody positive. So it's a problem that we're not going to wait out, that we have to be proactive. And so these therapies, like Robin mentioned, that where we can acutely lower antibodies and then pair that with an approach that actually prevents them from coming back is probably one of the ways that this could be managed. The antibody cleaving drugs are really fascinating. They're often human-specific enzymes, so it's been a little challenging to model those in animals, but it's possible that now to even eliminate IgM, the other form of antibody that causes some of the more significant safety concerns with these products, and as long as there's not a reaction against that enzyme, they should be very effective in managing both acute responses and those that have pre-existing antibodies. So they're both will address safety and ultimately, hopefully, improve durability and the level of expression. One of the things we've learned that's interesting about the antibody suppression technology is that antibodies are the principal means that the vector is eliminated from the bloodstream after they're administered. This starts at about 4 days after dosing, and then by a month is at its peak level, and that's what leads to ultimately the decline of the vector in the bloodstream. When the antibodies are not present, the vector persists in the bloodstream much longer, and those are in fact particles of the vector that carry the corrective dystrophin gene that can still be put to work. And so they increase expression levels in the absence of antibodies about 4-fold. So we're quite excited about that, even for commercial products, pairing these techniques with the opportunity to get higher levels of expression without changing the dose.
Yeah. I've seen you, there seems to be an approach here for a cocktail of events depending upon the individual. You know, I've seen you use rituximab, sirolimus, various other products in there in order to deplete or reduce antibodies. So there seems to be I think a combination of approaches that one could actually use in order to get patients down to a region where you might expect to be able to dose them.
Yeah. Well, again, in this kind of concept of repurposing, there are drugs that have been developed for hyper-IgG syndromes, one of which reduces broadly antibodies in patients with myasthenia gravis. And there are several products that are available, and we're studying 2 of them. And with the help of actually support through Cure Duchenne and other organizations that have a vested interest in this patient community, we have applied to the FDA to use this in an off-label fashion in boys who were either previously received gene therapy, where it's, I think, is not as likely to have a profound effect because it doesn't completely eliminate antibodies, but those that are very close to the threshold of allowability, this product is actually a portion of a natural IgG and doesn't have any immune reaction to itself for the most part. And so we think it could be very safely administered immediately before the gene therapy. And what was a little surprising to us in the preclinical models is not only did it lower antibodies, but it also, when given concurrently with the gene therapy and by shifting the dosing to before and during the gene therapy, it actually prevented the antibodies from appearing for the period of time that this other drug was administered. So that actually offers a great opportunity for enhancing safety because you'll unlink the peak of the vector product in the blood and the onset of antibodies. So we hope to be, in the next month, beginning further testing that product.
Okay. Robin, one thing that was recently published out of Sarepta, and I bring it up because it's certainly of general interest to the audience, is vector shedding. You know, there's families with multiple kids who want to go through this, and they're concerned that one would be dosed, there's a risk for the other one actually being contaminated. And I know the studies are quite clear now. You want to say a few words about what we know on vector shedding?
Yeah, sure. So in our 103 study, which is in humans, we looked at vector shedding. And what we found is that the vector shedding peaks in saliva and urine at day 1 after gene therapy administration, and then in the feces at 2 weeks after. And then we further found that after the peak, there is over a 99% reduction at 4 weeks for all of these samples, so in the urine and the stool, in the saliva. And so we do recommend the typical precautions that you would take for anything infectious like a cold or a virus, especially for the first 4 weeks. Use gloves, things like that. We've also looked at vector shedding in a mouse model, and so what we did there is to take mice that had never been exposed to our vector, and then we did a simulation of vector shedding, which was to expose them to the vector through their mucosal membranes, which in this case was the eye. And so we did not find that they seroconverted, and that remained true even when we used titers up to the highest that is shed by patients that have been administered gene therapy.
Yeah, that's been very valuable human data that's been collected by Sarepta, because we often see the scenario where in a family there might be 2 affected boys, and one is eligible by age and the other one's not.
Yeah, I bring it up because I know there's families in the audience with 2 boys and more, that have concerns about that. And over the years, there's been stories about one being dosed and the families being separated for a while. But this type of information really helps inform greatly, and reduces a lot of the concern that's in there. I want to bring Sharif in. I don't want to leave you sitting quiet, so I'll shift gears slightly, and talk about the vast majority of capsids that we use today are all natural. AAV8, 9, RH7, 4, 5. And there's challenges with each of those. There's advantages with some of them. But I think that we clearly are looking at a time whenever we can engineer into a virus new ability to actually target different types of cell. Certainly for the case of Duchenne, we'd want to de-target towards the liver and be able to improve the targeting that these AAV capsids have towards muscle. And that's got a number of real knock-on benefits, not just obviously being able to target muscle much more efficiently, but on the dose that one might use, the manufacturability of this, the safety margins. So do you want to talk about where the field is today and where it's shifting? And I'll bring Robin in because I know recently that Sarepta have licensed in the myo-AAVs from the Broad, and just to try and understand where they are with that, because I think that there's where the field may be shifting over the next months and years.
Absolutely. I think you covered it very well about the current status of the field using naturally occurring capsids. These are viruses that are found in nature. They are good delivery vehicles for delivering genes into the body. The main issue is that when you inject them into the bloodstream, majority of the virus gets sequestered in the liver. So there is a small percentage of the virus that gets to non-hepatic tissues other than liver, including the muscle. And that requires using extremely high doses of virus. That high doses have been associated with different sorts of toxicities, including liver toxicity, elevation of enzymes, complement activation, kidney injury. So there is definitely an unmet medical need to deliver genes more effectively to the target tissue. And what we have done is to use a structural biology information, basically knowing that what's the structure of these capsids and where are specific parts of the capsid that you can modify to give them new functions. And we developed this new class of capsids that we call myo-AAV class of capsids that can deliver genes much more effectively into both the skeletal muscle and heart. And more recently, we have developed the new generation that are actively also detargeted from the liver. So when you inject them systemically, they not only transfuse a skeletal muscle and heart, which much higher efficiency and deliver higher amount of transgene into those tissues, but also gives us higher safety margin in terms of exposure to the liver. I'm going to present some data for our Duchenne candidate, KT-809, later this afternoon at 2:30. And we have clearly shown that at low doses, dose of 40 to 13, we can express high levels of the microdystrophin transgene uniformly in multiple different skeletal muscles and in the cardiac muscle. I think looking at expression in the heart is extremely important. There has been not much information about expression of the transgenes in gene therapy in the heart in patients. And I'm going to present some data in terms of both capsid transduction into the heart, also activity of promoters that we're using. That we need to make sure that we not only express enough amount of protein in the skeletal muscle, but also in the heart, as obviously heart is a major affected organ in Duchenne. And it's very clearly shown from preclinical studies that the higher amount of microdystrophin specifically that you express in the muscle, it correlates with function. So basically, if you have enough of the microdystrophin transgene protein in the skeletal muscles, it improves the force of the muscle, it protects the muscle against damage. It's all about having enough of the protein, and delivery is the main issue, which we believe that we have a solution for.
Thank you. Raman, any update on what's going on with myo-AAV that Sarepta licensed in? Is there plans to move that forward into study?
The myo-AAV platform, what would that be?
Okay. Right, so I can acknowledge that we have licensed the myo-AAV from the Broad Institute, and this is definitely an area of ongoing research for us. We're always looking for new ways to build on what we've learned and make sure that we have the latest and greatest for our patients. However, that is the extent of what I can speak to with the myo-AAV work today. I can also add, though, that we are quite happy with the vector that we have. It has a very favorable safety profile, and we do see robust expression in the skeletal muscle and the heart. So we are quite happy with what we have, but we will continue, of course, to explore other avenues as time goes on.
Excellent. So as much as, to Sharif, as much as one can actually modify and adapt and change structure of the capsid to have different tropism, for whatever the target may be, is there ways to do that where you could reduce the impact of existing neutralizing antibodies for one that had been pre-dosed with a 9, an 8, or an RH74?
That's a great question. I think that's a huge problem, obviously, right? There would be a time that we would need redosing for patients that have received first-generation gene therapies, and it could be attacked from multiple different avenues. We talked about Vyvgart, plasmapheresis, IDES. Another way to do that is by modifying the capsid. It's basically making either enough modifications in the capsids that we currently have that they don't get cross-neutralized or get neutralized less effectively with the antibodies that are present in the serum, or engineer other serotypes that have less cross-reactivity with AAV9 or RH74. And that's an active area that we're pursuing. I think it would be very important to tackle this from multiple different aspects, both pharmacologically and also from change in the capsid that can evade neutralizing antibodies. I think the first step would be to show that you're able to those patients that have pre-existing immunity. That would be a lower bar, and then the next step is redosing, because the levels of antibodies after first injection is going to be extremely high.
Okay, I've seen the data on your current capsid, and it certainly looks quite differentiating. How far do you think we can actually take this into a new era where we can design individual AAVs that will do what we want to do at lower doses?
I think with consecutive rounds of engineering and evolution, you can increase the potency. At the same time, we believe that at some point you need to move the best capsids that you have forward. And we believe that at this point, we have achieved that bar, that our goal is to produce physiological levels. And by physiological level, I mean same number of molecules of microdystrophin as endogenous human dystrophin that's present in skeletal muscle and heart. And our non-human primate data shows that we're able to achieve that with a combination of the best capsids that we have evolved, and also a new promoter that we have designed to be able to express transgene not only in skeletal muscle, but also in heart in primates. So I think there could be room for improvement, but we are at a stage that we have made the decision what we have is good enough to move forward.
Okay, thank you. The reason for the questioning really is it comes from a bunch of stuff that's been said over the last couple of days. I think it's very clear that AAV drugs are incredibly expensive, but very expensive to make. And if one is able to increase the tropism by 3-fold, 5-fold, 10-fold, that means in a manufacturing batch or a manufacturing run, you're making that many more doses available for the same amount of money, bringing the cost down, making it much more competitive, and certainly leaning into the safety profile that these drugs should have. So I think that there's an enormous amount of effort that's really worth putting into the field to try and develop it into something that is making it much more affordable for all of these diseases.
Exactly. That's the other advantage, that if you can use much lower doses, it would cost less to generate the drug, the price could be lower. The main advantage that we see of using these caps is obviously lowering the dose to avoid any potential safety issues, improving efficacy to make sure that you're getting homogeneous expression in a skeletal muscle and heart. That's a topic that Barry and Kevin also brought up, that if you get patchy expression of the protein in specific parts of the muscle, not other parts, that is much more amenable to be less durable. So the goal is to have the most potent delivery vehicle that can give you enough levels of microdystrophin homogeneously in both the skeletal muscle and heart. And obviously, it has other advantages in terms of lower cost of goods, lower levels of material that you need to manufacture as well.
Okay. And we've got about 7 minutes left, and I want to change gears slightly. I know you're having a little bit of trouble with the system in hearing my voice, but I want to talk about the ability to combine therapies right now. And combination therapies are in vogue, and it's something that's real. We're witnessing this with Edgewise, who've got individuals in their trial that are post-gene therapy. And I think that this is really a topic that's going to happen more and more. We've got gavinostat approved that's out there that could be layered on top and various other things. And I know that Sarepta has published some work where you've looked at the MDX mouse and treatment with a PPMO and then on top of that with gene therapy. Do you want to comment a little bit about where the company's mind is and how that might sort of mature over the next few years?
Yes, so it's obviously an area of interest with all the new therapies coming out, as well as our therapies that both restore dystrophin. And so what we did with the MDX mouse is we gave sequential doses of our exon skipping therapy. And so after 3 doses, the animals were then given gene therapy administration. And so we found no new safety signals there, so that was well tolerated, so that part is very exciting. We also found that this did not interfere with the function of either of these medicines, so we saw continued protein expression of exon skip dystrophin, and then we also saw expression of the microdystrophin from the gene therapy. And so it's important to keep in mind that these 2 therapies do work at different parts of the cell, and their mechanisms do not interfere. And so when we looked more closely at the muscle, we also found that these were both in the membrane at the same time and didn't seem to be interfering with each other there either. So these results do suggest that we do not need to have a washout period of exon skipping therapy before patients receive gene therapy administration. So yep, this is an area of ongoing investigation, and we are looking forward to continuing to investigate it further. There are currently no head-to-head therapy evaluations for medicines that are outside of Sarepta, however.
Kevin, you want to add a bit?
Yeah, I'll just add, we've done some pilot experiments, I might have mentioned to somebody here, looking in the U7 approach similarly to use a version of microdystrophin to do direct comparison to see do we see added benefit in with our U7 full length and then the expression of the microdystrophin. This is analogous in some ways to the kind of studies that should be done for consideration of microdystrophin in a Becker phenotype as well. So it's important to sort out. And I'll say in a preliminary data, we don't see any difficulties. Scientifically, they should, all exon skipping there should be fundamentally at a molecular basis in the cell compatible with a gene therapy. I think the biggest challenge parents will face about this, of course, is will insurance pay for this?
Exactly.
After a $3 million dose with continuous therapies for continual cost of exon skipping therapies. That's a really kind of separate issue. I do know there are parents who are asking for access to those therapies again, and we're going to have to see how the insurance, the financial world responds to
I
this.
think parts of that are going to be driven by both the safety profile but the increased efficacy. And the argument is always going to be made there.
That's right, yeah.
Yeah.
You can really start to imagine, too, that products that work by different mechanisms will have an even greater additive effect. And so particularly in the approach like is being used by Edgewise with 506 to protect fast-twitch fibers that are more prominent in the younger boys. And with the advent of early diagnosis, you could see really a role for early use of a product like that combined with another modification.
I totally agree. I think that's a really innovative approach, and I'm really excited to see the data as it matures over the next year or two. And I think gavinostat in there as well with the mechanism of HDAC inhibitor, again, very complementary. Could well be something that's worth studying. We've got a couple of minutes. Karsten, any final comments from you?
No, I think combinatorial therapies is the way to go, and then this is a tough condition to treat, and there's not one solution to all of it. And it is a combination of gene-directed or genetically directed therapies and those that are muscle supportive. So we talked about therapies that promote muscle health, help regeneration, are counteractive against fibrosis, which is a big barrier to effective therapies, and inflammation. So all of these, I think, if they're kind of selected to attack different angles of the pathophysiology of the disease, from the genetic causation to the downstream effects in muscle, and they're kind of combined in an intelligent way so that they don't interfere with each other. So you don't want to do therapeutic approaches that could end up being competitive to each other. They need to be additive to each other. And that, I think, is the way to go and has to be the way to go. It is, of course, a nightmare from a drug development point of view, because but
Yeah, I agree.
talking about the future.
Yep. So in the last 1 minute and a bit, let me just sum up a couple of things. First of all, I'm delighted to have this panel of forward-looking thinkers here who are really not just looking at where we are today, but where we're moving to, both in the design of capsids, combination therapies, the safety of these drugs. The field is fairly mature, but we've still got a long way to go. And I'm really reassured that we've got, the support of companies in this here space and academics and real thoughtful individuals who are thinking about patients. I think that we're at a really interesting time. We're going to see data, not just from the gene therapy companies themselves, but gene therapy in combination with other things over the next year and 2 years that I think really will inform our thinking about the best ways to tackle this disease. And with new capsids on the line, I think that it will change the landscape over the next few years. So with that, just join me in thanking this panel for their thoughts and contributions in here today. Thank you.
All right. Whew. How we doing? What a great morning. How about all these speakers? Let's give them all a hand. It's fantastic information. All right, we're about to embark on lunch. There's a Navigating Adolescence in this room at 12:45, so if you want your lunch, you can go get it and come back here if you're attending that session. Lunch is outside the doors like it was previously. Exhibitor Showcase. If you haven't been over there, spend some time. It closes down at 2 o'clock today. And if you are going to Discovery Cove, over where the swag bags are, between now and 1:30, you can pick up your passes, tickets for Discovery Cove. That sounds really cool. I think I'm going. Anyway, again, thank you. Go have lunch. Come back to the sessions. We will see you soon. Thank you.